Thanks for joining us. Back with Paleo Talks this week, we have Cristina Barron Ortiz. Very happy to have you. How are you doing, Cristina?
I'm doing good. Thank you, Blaine. I'm happy to be here with you guys, and I'm happy to be here with the audience, everyone that's watching from home, from the lab, universities, wherever you are. Yeah.
All over the world. And Chris, you can see he's drooling. He's over there thinking about the mammoths and the mastodons, and he's happy we're having another mammoth talk today.
It was not my devious plan at all, but it just worked that way.
And David, we'll go over you for a bit to talk about the show and how it works.
Absolutely. We're going to stick to our typical schedule. We're going to spend the bulk of the program with our guest giving a talk today about mammoths, like Blaine said. And then once the presentation is done, we're going to open it up to questions from the audience. So as the presentation winds down, we'll remind you to go ahead and leave your questions in the comments of the Facebook comment section of the video. And the end of the program will just be Q&A with our guest from the audience. And as always, if for any reason you can't leave a comment on Facebook, I'll be keeping an eye on the Gray Fossil site, Twitter and Instagram accounts, so you can send us a message there as well. Now, before we get started, I want to tell a very short anecdote, and this I think will make Blaine very happy. The other day, I went to the grocery store to pick up groceries, and the woman who came out of the store to give me my groceries saw my name on the page and said, "Do you do Paleo Talks?" And I said, "Yeah." She said that she watches Paleo Talks. So hello to that person who recognized me at the grocery store. Thanks for watching.
Fantastic. That's a first for sure. And we're on episode 41. Is that right?
That's right.
Yeah. Incredible. Incredible. And it was shortly after this time last year, that we started. So we'll continue going. And again, we're being brought to you by the Center of Excellence in Paleontology at East Tennessee State University here in Johnson City, which also has the extraordinary Gray Fossil site, this place that everybody should come and visit when you get the chance. 5 million year old locality. We don't have mammoths, but we do have mastodons. And it'll just blow your mind. Come visit us when you can. And I'm Blaine Schubert, and let's just move forward and hear about Cristina. And Cristina, what we usually do is we have our guest tell more about your background and how you got into paleontology in the first place. So what interested you when you were younger?
Yeah, so I'm from Mexico. So I was born in a town that's called Zacatecas in central Mexico. And since I was a little kid, I remember being fascinated by dinosaurs and prehistoric animals. When I was 12 years old, my father got accepted to do a PhD in the United States. So we went to Amherst, New York. So he went to University at Buffalo, SUNY at Buffalo. And at the time, I remember that first summer we were there, I was just playing around in the backyard of our house, and I picked up a few rocks. And later, the following day, actually, I was looking at them, and one of those had a tiny fossil. It was a brachiopod, quite small, very small. And I still have that fossil at home, well, with my parents in Mexico. And that's what really got me hooked into paleontology. At that point, I told my ESL teacher about it because I was going to summer camp for ESL at the time. And then she told my science teacher, 8th grade science teacher, who's gonna be my teacher for the next year. And he approached me that first day of class, after class, and told me, "So I heard that you like fossils." He was a geologist by training. So Mr. Shalacy, he passed away a year and a half ago or so. But he was the best mentor that I've had. So he gave me all of his college books. They were from the 1950s, of course, but they were still, I was still fascinated by them. So he allowed me to take one of the microscopes from school, take home and look at the fossils that I had and that I was finding. And we also went on fossil collecting trips. So that was, that's how I got started into, like, really got hooked into paleontology. And my parents also really encouraged my interest. So, yeah.
And where did you go to university?
I went to, so we went back. We lived in the United States for 4 years. Then I went back to finish high school in the U.S. Mexico, and that's when I got hooked on the quaternary, because one of my uncles has a farm, and close to what his farm is, they were finding some mammoth bones, and some camel bones and horse bones, and so I went to take a look there with my cousin. So that's what brought me to the near time, I guess, into the quaternary from the Paleozoic. And I went to the university in Aguascalientes to study biology. So that's in a city of Mexico too. And then I moved to Canada, to Calgary in 2009 to do a master's, which then turned into a PhD. And then I started working at the museum in Edmonton in 2015. And I've been here since then. Yeah, so it's gonna be 6 years, almost. Yeah. So that's my journey in as far as how I got to where I'm at right now.
Wonderful. I love how you said hooked on the quaternary. I think that's a really deep set hook that many of us get. You can't really get out of that once that hook gets you.
No, I think the quaternary is quite interesting because it bridges the past and the present. It's like and it overlaps with a lot of disciplines like archaeology, archaeology, and yeah, I'm collaborating with some colleagues on looking at and domestic horses too, which is quite cool. Yeah.
Wonderful. All right. Well, thank you for sharing that history.
Thank you. Yeah.
Do you want to go ahead and share your screen?
Yes. Let's do this.
I think the quaternary is what we call a treble hook. It's one of those 3-prong hooks.
Mm-hmm. Why is that? I guess it's.
Oh, I just mean that you really you get hooked, and you can't really escape that hook. You're set for life when you fall in love with the quaternary.
Yes. Yeah, I agree. Okay. Can I start now?
Yes, please do.
Yes. Okay. So the title here of my presentation is Mammoths on the Move: Understanding the Fossil Records of Mammoths in Alberta, Canada. And right off the bat, so this is a project that I'm working on in collaboration with Chris Jazz from here, also from the Royal, and from Tasha Kamich. She's my former lab mate from the University of Calgary. And right now she's working at Caltech in the United States. So yeah, this is a project we're working on. Blaine, I just want to ask you a quick question, because on your screen, is our face covering part of the slide, or is
yes.
It is? Should I move?
Yeah.
Oh, I can move us.
It looks perfect. I can just see the mammoths.
Oh, okay. I wasn't sure if you can, because I can see you and myself.
Oh, I can also see you over in the corner as well, but the screen
it doesn't really
is good.
It doesn't cover the slide. Okay. Yeah. So here in North America, for the Pleistocene, different species of mammoths have been named over the years. But in recent, most current researchers identify at least 2 species for mainland North America, or types of Colombian here on the left, and the woolly mammoth here on the right. And they were the woolly mammoth, of course, is probably one of the most iconic animals of the Ice Age. That's what usually people think about when they think about the Ice Age. They have this long woolly coat. They were shorter than the Colombian mammoth, a bit smaller, well, smaller than the Colombian mammoths. And also their distribution, as you will see, I have a map. Woolly mammoths were distributed more in northern latitudes, and the Colombian mammoths were distributed in more southern latitudes. And there's been a lot of more recent exciting research going on about their relationships and the genetic aspects of them. So there is a recent paper that is suggesting that the Colombian mammoth is a hybrid species. We also have some evidence hybrid species of hybrid between the woolly lineage and another lineage that morphologically appears to be similar to the steppe mammoth. There's also other evidence of hybridization during the late Pleistocene between the Colombians and the woolly mammoths. So all of this is introducing more complexity about their boundaries as species, and also about complexities about understanding their relationships, as well as like the morphological aspect of them. As I mentioned before, so the woolly mammoths are distributed in northern latitudes. This map I just took from Neotoma, the Neotoma database. It's not comprehensive because, of course, if you look further south into Mexico, there's not a single dot, and that's because this database started with the United States. And yeah, Colombian mammoths extended further south into Central America. But even though it's not comprehensive, I wanted to show you this map just to highlight that difference in their distribution, where the woolly mammoths indicated by the red triangles have this northern distribution, and the Colombian mammoths indicated by the green circles are distributed more in lower latitudes of North America. And so the area that is now Alberta, which I'm highlighting here on the map, that area is, as you can see, it's located in that where traditionally that the area of overlap for these two types of mammoths. So, I mean, woolly mammoths extended further south, but as far as Colombian morphology, that's about the limit of the range. There's a few specimens from up in the Yukon that have a morphology similar to the Colombian mammoths. But I know some researchers like Lister and Scher, they identify those as Strogonterae, so the steppe mammoth, and that's because, well, Colombian mammoths, as far as the morphology of their teeth, and the steppe mammoth are quite similar, so they do overlap a lot morphologically. But the genetics is allowing us to provide a different perspective into the relationships.
It does look like there's a green dot up there in the.
It is actually, yes. So there's it's behind. It's behind the red triangle, and yeah, in Neotoma, that dot is identified as Mammothus columbi. But I know, that in Lister and Scher, in their publication in 2015, they have that specimens from the Old Crow area and other parts of the Yukon and Alaska as Mammothus strogonterae, the steppe mammoth. Anyway, so going back to Alberta, so this area is interesting too because we would expect, like over the history of throughout the Quaternary, we would expect as environmental changes happened in this part of North America, that the ranges for these animals would shift up and down, and that populations might move further north of populations with like a morphology of Colombian mammoths, and populations of woolies would shift further south. And this is a working hypothesis because there's evidence for other animals about this complex biogeographic dynamics that are happening during the Quaternary. So the area that is now Alberta is at times formed part of this passageway that connected Beringia to the north and the rest of the continent to the south. And before we went on live, Chris Whitgen and I were talking about the mastodons. So there's a recent study that was published last year by Krapinski et al., looking at mastodons from different parts of North America, looking at the DNA. And their study seems to suggest that during interglacial periods, so during warmer times, as more forested environments move further north and even into Beringia, that mastodons extended their distribution further north. But as the climate got colder and drier, and those forested environments started to disappear, some of those mammoths, some of those populations were locally extinct. A few of those may have survived for a little bit further north in Beringia and a few refugia, but eventually it appears that the climate got so cold that those forested environments just weren't there as we were approaching the last glacial maximum. And so there's this clearly shows that there's this complex and dynamic aspects with the distribution of the mastodons in this case, of their biogeography. We have seen similar things for other animals like bison, for example, that after the last glacial maximum, after the ice started to melt and there was this corridor forming, that populations from the north started to move dispersed further south into what is now Alberta, and populations from the south started to disperse north. So that here in Alberta, during the post-LGM, so the post-glacial, so about like 14,000 years ago, we started to see show up in the record animals with genetic affinities to the north and genetic affinities to the south. So oh yeah, and this area has also been suggested as one of the potential routes by which people dispersed from Asia into Beringia and then into North America. The other route, of course, being the coastal route. And there's a lot of research going on about this specific issue. Some people really favor the coastal route because it appears based on the timing that we see people further south in North America and into South America that maybe that central route, the corridor route, was closed. But anyway, but there's still a lot of research going on also in that topic, and that's why this area is also interesting because of that dispersal of people. So that passageway was relevant not only for animals but also for peoples in the past. And so that's why in our study we wanted to see at the morphology specifically. So it's a morphological study, the one that I'm going to talk about today, and look at how the woolly morphologies and the Colombian morphologies are distributed in Alberta and in time and space. So this is an ongoing project. So I'll show you some of the results we have for the morphological analysis, and we have some results for some dates, but we still need to get a few more dates on the specimens we looked at. But yeah, our hypothesis is that we will see these shifts in the distribution of the woollies and Columbian morphologies through time and space in what is now Alberta. And so mammoth remains here in Alberta. So in the map on the left shows these are proboscidean localities for Alberta, for the province. Most of these localities are for mammoths. There's very few mastodon sites and mastodon specimens. So yeah, so most of those dots are for mammoth remains. A lot of the remains here that we find here in Alberta are quite fragmentary. So we find a lot of isolated teeth, isolated vertebrae, and portions of tusks, for example. And that's because a lot of the specimens are found from fluvial deposits, from these high energy deposits, especially sands and gravels. And a lot of them have been found as industry, sand and gravel industry, as they're extracting and putting blades in the ground to extract all of this material, they come across fossils. And Chris, my supervisor, and our predecessors still here at the museum have built a strong relationship with the sand and gravel industry, that they typically contact us whenever they find some bones, and then we can go out there, collect them, and see if they're in section. We can see them in place. Although in these high energy environments, a lot of the specimens sometimes are reworked, and that's why they're quite beat up and fragmented. But
so Christina, are these glacial outwash deposits then?
No, well, there's some glacial outwash deposits. The one here shown in this picture, this was a valley fill. So it was this was pre-LGM, before the last glacial maximum. So it's around the Edmonton area. So there was it was part of the old North Saskatchewan River, or where the old course for the river. Yes, and oh yeah. So here's a picture. This is, of course, it's not a mammoth specimen. It's part of a bison forecore. But I wanted to show you this picture just to show you how we typically see them on site. This is at a gravel pit in northern Alberta. And so yeah, so a lot of those specimens are quite beat up, but nonetheless, there's still a lot of information we can get from those specimens. From other studies, we know that DNA preserves well, And also we can, depending on how complete they are, we can do some morphological analysis.
Yeah, looking at those gravels and how rounded they are, it's amazing you have anything left.
Exactly. Yeah, this particular specimen, I was surprised that it was like that it survived. Because yeah, looking at that, I would think that it would get just destroyed. Yeah. Occasionally, we do find more complete specimens. Rarely, but they do exist. Like this juvenile mammoth mandible that also came from those fluvial deposits. And also we have one associated specimen. So this is with the most complete mammoth specimen we have in the collections. So it was found before our time, before we started working here at the museum in the 90s. So our predecessors were notified by the gravel pit company that they had found some bones. So when they went out, they didn't find them in place. They had already, a lot of them had already been extracted. But based on the elements that are preserved, based on the fact that they were so close together in that area of the gravel pit, and also based on the fusion of the bones, it appears to be that they're all from the same animal. So this is so far the most complete specimen we have here that has come out of Alberta. But yeah, like I said, a lot of the specimens are fragmentary. We have several teeth, and that's because, well, these teeth are hard and durable, and they preserve well. And for this talk, and also they're relevant because the distinction between the different types of mammoths has relied heavily on the morphology of the teeth. And so it's good that we have some teeth to study. This one here is an upper tooth, an upper M3, so the last molar to come out. And shown here in this diagram is the diagram for a lower M2, a lower second molar. And I just want to put out this diagram to show you the main features of mammoth teeth. So those numbers are what are called plates or lamellae. So yeah, mammoth teeth have an elephant teeth to have this series of plates on the teeth. And the picture on the right is showing the tooth from the occlusal surface. So the surface that the chewing surface. So yeah, that's one plate. Then there's the enamel. So all of those loops are enamel bands. On the inside of the loops is the dentin, and then on the outside we have cementum surrounding those enamel plates. And mammoths as well as elephants are quite cool because of the way that their teeth erupt and how the teeth they have. So I love this picture. This is a specimen. This is a picture that I took when I was visiting the Natural History Museum in Paris, France. And it's a wonderful specimen because it's a modern Asian elephant. Is the head. So the front part of the head is to the left, and the back of the head is to the right. And then we have a cross section across the head, and we can see the teeth, and we can see how we can see the last premolar and the first molar. And in mammoth as well as elephants, the teeth were being replaced as the front teeth was being worn. There was a new coming behind it and just pushing it forward and being replaced. So they're pretty strange in that sense. They're quite unique. And so yeah, so for like I mentioned before, so the teeth have been used to separate the different types of mammoths. And so one of those features that researchers have looked at is the number of enamel plates. So woolies, shown here on the right, have more of those enamel plates than Colombian mammoths on average. Also, the spacing of the plates in the woolies, they're more tightly packed than in the Colombian mammoths. And the thickness of the enamel is another feature, so that in the woolies, they have thinner enamel bands than in the Colombians on average. All of this is on average, because they as you'll see, they overlap quite a bit. In our analysis, what we looked at, well, I guess I'll show you the question. So what we're interested in our studies to look at how much variation is in the teeth that we're finding here in Alberta. Do we have only woolly morphologies? We have Colombian morphologies. Are there any like intermediate morphologies of the two? And also we want to know how they are distributed in time and space. Are there any differences between woollies and Columbi morphologies? Like, do they coexist at some places? Are they like contemporaneous, or is the distribution disjunct geographically and in time? And so the measurements we looked at, so a lot of our specimens are not complete, or they weren't beyond the point where you can estimate the total number of plates in an unworn tooth. So we couldn't really do the plate count. So the next two things you can look at is the spacing of the enamel. So we do that by looking at something that's called lamellar length, which is length between one plate and one cementum interval. And you calculate that through different measurements on the teeth. And then the other thing we looked at was the enamel thickness, measured at different points of the tooth. And we also measure the width, the maximum width of the tooth, because with the width, we could standardize the measurements and calculate some indices. So the lamellar length index and the enamel thickness index, those are standardized to a tooth size of width of 100 millimeters. And standardizing the measurements allows for more consistent comparisons between teeth of different size. And so for our analysis, that's what we looked at specifically. So we looked at the lamellar length index and the enamel thickness index. And we use data sets that have been published, including the data set of Lister and Scher from 2015, and the data set by Whitguy et al. From 2017. And in this talk, I will show you the results of the data set from Lister and Scher. And so I'm gonna walk you through the results just because this graph is gonna get messy. And so I'm gonna slowly be adding the points, the data points. So shown on the x-axis, so the horizontal axis, we have the lamellar length index, so how the relative spacing or the relative width of the plate and cementum interval. And so on the left side, so woollies in this case, they have this, they're shorter. And I put the Colombians here. And for Colombian morphologies that have this wider lamellas. And then on the other axis, we have the enamel thickness. So that is thinner towards the bottom, thicker towards the top. So that, yeah, woollies tend to be on average, have these thinner enamel, and the Colombians have this thicker enamel. So the dots here, so the dots for the woolly specimens are from Varangia and Russia for the late Pleistocene. The dots for the Columbi specimens are from the middle and early Pleistocene. And now if we plot the late Pleistocene specimens that have been identified as Columbi from the Colorado and the Rocky Mountain areas, this is where they fall, the crosses. And then looking at these mammoth specimens from the Midwest and the Great Lakes, here they are, the triangles. So they overlap, they basically span the whole range. And that makes sense because there's evidence that some of those is like the woolly morphology, and we have the Columbi morphology, and yeah, it spans the whole range in that geographic area. And then here are a few specimens from the Southwest, the stars, and Mexico too. And then the dashes are specimens from the Southeast of the United States. A lot of them are from Florida. So it is quite a messy graph. As you can see, there's quite a lot of overlap between the woolly morphology and the Columbi morphology. Shown in the shaded area is the area of overlap between the northernmost specimens, so the specimens from Varangia, and the southernmost specimens, so in this case, specimens from the Southwest. And shown here, these are the specimens from Alberta for the upper third molars, the last molars. So it is interesting that we have, it spans quite a bit of the range two, but for the upper teeth, we have these two separate clusters. So we have the cluster on the right, we have specimens that have the columbi morphology, and then the cluster on the left, we have specimens that have more of the woolly morphology. So that's for the upper teeth. And then for the lower teeth, once again, so these are, the dots are woolly specimens from Beringia and parts of Russia too. Then we have specimens from early and middle Pleistocene of columbi morphology. Then specimens from Colorado and Wyoming, I think. And then the Southwest and Great Lakes area, the Southwest of the United States and Mexico, and the Southeastern United States. And once again, that's the area of overlap between the northernmost specimens from Beringia and the southernmost. In this case, they're from the Southeast, from Florida. And here are the specimens from Alberta. So here, unlike the upper teeth, we see a gradient that spans from the woolly morphology on one side to the columbi morphology on the right side. And we have a couple of specimens that fall in that area of overlap. That in our manuscript, we're calling those specimens with intermediate morphologies, but we don't make any assumptions about the relationships, because we really need independent data, like DNA data, for example, to try to make more sense about, are those maybe hybrid specimens per se, like proper, or what are they like, or are they, yeah. So in this case, we're just calling them intermediate mammoths with intermediate morphologies. Anyway, so those are the results for it as far as how they plot and in morphologically. I want to show you in terms of space. So this is again from the Neotoma database. The dots that you see are from specimens that we didn't look at, well, with the exception of, I don't know if you can see my cursor, the arrow here, with the exception of the green dot in the southeast of the province. That specimen is from, it's a tooth, it's a lower M3, but the other ones are not M3 specimens, so that's why we didn't look at those particular specimens. The red triangles, and we feel we're unsure about the IDs of some of these specimens too that are on Neotoma, because the southernmost red triangle in Alberta, those are mammoth footprints from a site that is called Wallace Beach, and they have been identified as woolly footprints, but just based on the fact that there's a few specimens, like the specimen from Calgary that Harrington and Shackleton identified as a woolly. It's a second molar, but it hasn't really been dated. So anyway, those footprints, they could be woolies or columbi. I guess the size might tell you. Anyway, but okay, I just want to plot here in this map the specimens we looked at. So shown here are the woolly specimens in our samples. So some from the southeast of the province around the Medicine Hat area, and other specimens from the Edmonton area. And then for the columbi, specimens with columbi morphology, here they are. So we have, again, some from the southeast part of the province, from the Edmonton area, and there's one specimen from the northwest part of the area, close to Grand Prairie, that town. So at least morphologically, we have some of the northernmost specimens that are known for the columbi morphology here in Alberta. There's also a northern one from British Columbia, from BC, that is plotted in Neotoma. But yeah, so that is quite interesting that we see some of these morphologies going quite far north. I admit that we don't know a lot more about the north part of the province, just because we don't have a lot of specimens from that area. Part of that is because there's the boreal forest that covers a lot of that land. Also, part of that is that because there's not a lot of people living over there that can come across fossils, and also I guess we haven't really done a lot of field work up there too. But oh yeah, and then the next one, yeah, those intermediate specimens are here from the Edmonton area. That's the square, the blue square are those intermediate specimens. So So as far as their age is concerned, so there's been a few published radiocarbon dates for some of the specimens we looked at. I'll show you those here now. Unfortunate, some of those are infinite. So we have, for the Edmonton area, we have infinite dates for woolly morphologies and infinite for columbine morphologies. And one of the intermediates has been dated to 43,300 plus or minus 3,000 years. So it's quite a bit of that. The standard deviation is quite big, so it's quite large. So, which makes me think that maybe we should predate that specimen. Actually, that specimen is actually that partial skeleton that I showed you before. That's one of those intermediate morphologies based on the teeth, of course, looking at the tooth. And then the other date we have available at the time is for specimen from the southeast of the province. It's actually, this is a specimen that was published by Churcher in 1972, and then dated by Hills and Harrington in 2003. And the date came back as 10,130 years old. Oh yeah, this specimen also was included in the study by Enck et al. In 2016. So there's some DNA data for that particular specimen. Yes. And so, yeah, we definitely, that's the next step for us too, is to, well, try to get some funding to date more specimens and see how their distribution, their patterns in time. Right now, with the data that we have, at the very least, we can say that during the post-glacial, so after the last glacial maximum, which happened about like between, I don't know, 20,000 and 15,000 years ago or so, during the post-LGM, at least we can say that mammoths with columbine morphology, some populations came back to Alberta as the ice was melting. Maybe some woolly specimens came back, or animals, but we don't have any directly dated specimen that's consistently identified as woolly. But yeah, so there's and this is what we can say as far as them coming back to the province after the last glacial maximum. Although they didn't come back for long, because at around 11,000 years ago, a lot of these animals here in Alberta and other parts of North America went extinct, so they disappeared, leaving behind the remains. This actually shown here in the slide, these are footprints from that Wallace Beach site in the south part of the province. And we've done some recent fieldwork in that area over the last few years in collaboration with colleagues from Archaeology here at the museum where we work, Chris, Jazz, and the Ecological Survey, and members of the Ecological Society of Alberta, and colleagues from the what is the National History Museum in Ottawa, the Canadian National History Museum. Anyway, but yeah, I've seen those footprints in field, and it's quite interesting because they're quite. It's amazing to see them on site, because you're immediately looking around to see if you can see the mammoth that left them, because some of them look so fresh. Yeah. Anyway, so we still have to do more work. Hopefully, as we continue to do field work, we can find more specimens. At Wallace Beach specifically, we have footprints, but we don't have bones so far of mammoths. So we're looking. And also, hopefully, as the sand and gravel industry continues to work, more specimens can be found that we can also study. And the next step, of course, would be to try to look at the genetics of some of these specimens. Right now, our study is solely morphological, to kind of build a framework, a better sense of, like, morphologically, how are the mammoth teeth that we looked at from Alberta, how are they distributed? But of course, the next step is to look at the genetics and see how, well, yeah, what that tells us about them, and in the bigger picture of what was going on in North America.
Christina, what do we know right now about the genetic work on mammoths? If you could tell the audience, you know, is it that we still think that there were 2 or 3 species, or is it more toward the one species hypothesis that's prominent?
I don't know, actually.
Okay.
Well, the thing is that there's evidence of that interbreeding between those Columbi mammoths and woolly mammoths. And like we need to, because I know I'm I mean, it is a fine line about how do you define a species, right? Like how much, because even modern animals, sometimes you have species that are interbreeding. And some of those hybrids are crossing back to their parent populations, and you have that admixture and introgression. But you don't have, so that's how you have that gene flow going to the parent populations. But still the parent populations could maintain some degree of separate identities and be called different species. It's all like a gradient as far as how much of that interbreeding is going on between the two types of mammoths in this case. And I don't know, I guess I could ask Chris Witka, because as far as I don't know how much evidence of like
Yeah, what are your thoughts, Chris, on this?
like I guess my question is like how much is this interbreeding, like is it like how did it happen a few times in the past, or is it something that like was constantly happening in their area of overlap? For example, here in Alberta nowadays we have hybrid zones, especially for birds, for example, and other animals too. And actually when we started doing this work, because the genetic work had already been published, Chris and I were talking, and Tasha too, we were talking about, so what if all of our specimens are like intermediate? And maybe Alberta was a hybrid zone for mammoths in the past. And so that's one of the things we had in mind as we were looking at the morphology. And yeah, so I wouldn't, yeah, what are your thoughts, Chris? Witka, yeah.
But I think we would agree on a lot of things. And I think there's some scale issues too. So with this most recent paper with the million-year-old mammoth DNA
Yes.
in Siberia, you know, that it kind of suggests that we do have this hybrid speciation event sometime deep in the mammoth family tree,
Right.
where you have woolies and steppe mammoths, maybe some unknown species of mammoths that there is some sort of hybridization event that happens way deep in the past, and then that turns into Columbian mammoths eventually. But on the other hand, you know, when we have looked at kind of the narrower scale, where we're looking at you know, contemporary mammoths in Alberta that show different morphologies or overlapping morphologies, genetically, at least in terms of the mitochondrial DNA, they aren't as distinct often. You know, so in that case, we may have we may be seeing a local population that is more variable because of closely related parent populations. So yeah, it's fun. I know when we were writing up some of this stuff, you know, the co-authors would say, well, do we synonymize things and make them all woolly mammoths? And you know, as a morphologist, I say, well, I really don't want to do that. But there's also some clear genetic patterns that you have to kind of account for as well. So
Right.
they're really fun to think about. Yeah, this is a textbook case of pick your species definition in some ways.
Actually, I was going to say that until recently, because now that we can get DNA for all of these animals, and even DNA going back all the way back to like 1 million years old, that's a recent development. In the past, the way we were defining species was through their morphology, and the way that the mammoth species have been defined, and the holotypes, like the holotypes of Mammoths columbi, it's their teeth. And the morphological characters are the ones that are the diagnostic characters that define what Mammoths columbi is. So we gotta make sense, like the genetics is telling us something, but we need to bridge that gap. Like we're probably the first generation that is encountering to this great definition of like, how do we define the species? Because we can do it have a phylogenetic species concept, for example, where we have the genetics and look at the tree and decide, okay, at this point in the tree, all of these populations are called woolly columbine, or whatever we want to call them at this point in the tree. Or we can also, sorry, yes.
Well, I don't mean to interrupt, but I mean, at some level, it also comes back to how we define those morphological species. We're left with teeth for most of these specimens. And, you know, is that because our characters just don't give us a particularly good resolution? They are variable characters within these different populations, and they show some overlap. You know, Alberta is this super highway for mammoths and different populations of different things at different times. And so you have this potential for a kind of glommed together paleontological record where you're conflating space and time and different biological populations and things like that, and they appear side by side. So, you know, going back and getting lots of radiocarbon dates and things like that, and defining, you know, where those lines are, where those chronological lines are, and where getting at potentially populations would be really fun.
So the really fun project that you guys are probably already doing, but would be just to look at all the different morphological variations across the spectrum and then do DNA on each and see how messed up all of that is. Because I can imagine that the morphology is not necessarily going to lump things into the more closer related clusters together. And if that's the case, it gives us this other sort of warning sign about our interpretations of morphology indicating closer relatedness.
Christina, I'm really excited that you're working on mammoths because you started with horses. And
which is even more difficult.
Horses are so messy and noisy that, you know, bringing that perspective to mammoths, I mean, how is that transition for you? I mean, is this kind of natural for you to think about this sort of variability?
About, like the variability, well, you know, it is, well, I guess to be honest, my first impression when I was like looking at the mammoth teeth is that the characters that are used are there's very few characters, but I think it's because of the nature of the record. Because as you mentioned, a lot of the ideas are based on the teeth, and within the teeth, there's really only 3 characters that are used that might be like relevant, and that is I guess we can say 4. For the history of mammoths, that's the height of the tooth, hypsodonty. Then you have the number of enamel plates, the spacing, how packed these plates are, and the enamel thickness. So to be honest, I was thinking like, oh my God, that's so few characters to actually like make sense of this, especially because these characters are tied to feeding ecology. They're all completely tied to demands on the dentition from their diet. So the tooth height, of course, more abrasive either from the plants or from the environment. There's a selective pressure of more abrasiveness happening in the teeth. The addition of the enamel plates, that's because you need to be more efficient at processing food, low quality food, that is tough vegetation. You need to be able to process more of that in less amount of time. You need to be more efficient at grinding that. So to be honest, I'm like, those characters are, there's no way that the genetics and the morphology are gonna match up, just because of the way that I see the morphology of the teeth tells me the history of their selection, that these animals have encountered selection for feeding challenges in their diets. Now they're not, I think we were talking a little bit about this before we went on live, because the way that I see is like some of those changes, once you have them, they're good to process some top vegetation, but you can also process maybe other plants. What I'm trying to say is that maybe you might be seeing something like Dollo's law here, the irreversibility of evolution. So once you have evolved high crown teeth, you cannot go back to low crown teeth as like to the same degree as you started, even if you're eating, even if you're browsing, or I guess unless that's really damaging your dentition. Yeah.
Well, it's a great animal to be studying sort of as a case study for sure, and looking at the comparison of morphology and genetics. And your preservation up there is quite extraordinary too, isn't it? The level of the collagen preservation and the protein preservations.
Yes. Oh, yes. I was just gonna mention it because I want to bring back to those horses, because in horses,
Sure.
at least there's a few more characters. Like there's the metapodials that people have looked at. There's the dentition, of course, and you may have also the same issues with like selection on the dentition. That's like the feeding ecology affecting their morphology. But I think it would be really good. I mean, we have to look at expand for mammoths, expand, and I know some researchers are looking at this to expand, increase our morphological data set from us in addition to the teeth. What are your thoughts on that, Chris? Like,
I think that's a great idea. But you know, like you said, it's you're limited by your preservation, and teeth are common, and you know, sometimes it comes back to, well, can we think about these teeth in different ways? I think number one, it'd be nice to have some nuclear DNA on these things. A lot of the genetic work in North American mammoths and kind of looking at this Columbian woolly mammoth separation is done with mitochondrial DNA. So it'd be nice to kind of revisit some of these things with new techniques and that can get at nuclear DNA. But then, you know, there are things about mammoth teeth that we haven't necessarily figured out how to measure very well.
right, they're hard. Yeah. Like
Yeah.
what?
Well, I mean, like for instance, the enamel. How much enamel volumetrically do we have in a tooth? You know, is that something that would play out in space and time?
Oh, okay.
So you know, there are fun things to think about yet with mammoths, I think.
Yeah, you're right. Because I mentioned those 4 traits. Those are the 4 characteristics that people traditionally have looked at. That the hypsilonty, the lamellar frequency or lamellar length, yeah, the thickness. But yeah, you're right. There's other ways. I guess it's an opportunity to be creative too, and figure out how else we can study them. What other morphological information there is that we can gain from it. And you're right. Maybe that the volume of enamel might be.
I think we better go to some questions from the audience. David, do you have one ready?
Yeah, we've got a few questions. So reminder for people, if you haven't already, leave a question in the comments and we'll see how many we can get through. We've got a few already. Let's start with this one from Aaron. Aaron says, based on the tooth morphology between the 2 species, can you tell how their feeding ecologies might have differed?
Based on their I mean, I can tell you that traditionally, what people thought about what they were eating. So like for example, woolly mammoths, they were proposed to have been, I mean, the more we learn about ecology and reconstruct their diets, the more we realize that they're quite flexible. A lot of these animals, they're eating a variety of things. Like I've done studies, dietary studies on horses, and you see some mixed feeding on horses in like places in horses. From the Yucatan, for example, where you had yeah, so it's sorry, what was the question? Whether I can tell them, like tell exactly?
The question was, using the tooth morphology, can you tell if there were differences in their feeding ecology?
To some extent, I guess you kind of enforce it, but no, actually I would have to. We have techniques for looking at like microair, mesoair, isotopes. Those are way better indicators of what the animal was actually eating, a particular animal. Yeah, there's been selection for for that morphology in the past, but that morphology is, yeah, like I said, the more we find, the more we realize that animals have a lot of animals have this flexible dietary flexibility, dietary plasticity. Yes.
Okay.
Mhm.
Well, another question on a similar line with a different method. Grant Boardman has chimed in with, as he puts it, his requisite isotopic question. Grant always likes to ask his questions about isotopes. This time, he says, do stable isotopes differentiate the ecology between the species?
If they differentiate, when you say that between like woolies and columbi?
Yes, I assume that that's what
Yeah.
he's asking.
Actually, no, I don't know for that one. I know there's a lot of isotope studies that have been done. I mean, I'm trying to think. From what I know, as far as like, well, the animals, the woolies that were living in Beringia, are different from what I know of those environments. Those environments were sometimes extremely dry, and I truly believe that they only had like grasses and forbs, like really short, low shrubs to feed on. So if you look at the isotopes, you would tell that difference between those populations living in the north and then populations living in Mexico, for example. Yeah, so.
I can jump in there a little bit too.
Yes.
So, we're kind of dealing with an evolutionary time scale and an ecological time scale. And so
Right.
tooth morphology is really evolving to address these long-term trends in diet. Whereas things like isotopes, things like microwear, they're really addressing the diet of that animal as it lived. And so, places like Alberta where this forage changes fairly rapidly and things like that, that ecological timescale can change very rapidly, and it takes a long time for the evolutionary timescale to pick up and to catch up. So yeah, these are fun kind of tensions within paleontology. I think that is one of the really enjoyable things about working with proboscideans.
Right. Can I add something? You're right about the time scale aspect, because yeah, the morphology, it's more evolutionary at that scale. So it's longer, so you need to have that selection pressure to over the generations of animals. Yeah. I know that in the traditional sense, like before traditionally, mammoths were viewed as this anagenetic sequence, or like chronospecies, where you have this trend in some of those morphological characters. Do you think, Chris, I'm asking a question now instead of answering questions, because the way that I see it is that maybe the selection pressure was more episodic than what we think. That although it was for a longer period of time than ecological time, it wasn't like a constant selecting pressure to in one direction, for example. Yeah.
Yeah, I mean, I'll give a short answer. I don't know. There's so much biogeographic kind of variability going on that it's going to be really difficult to sort that out. And I don't know that we have the data right now or the chronological resolution to really address that, at least in North America.
Right. Yeah.
David,
All right,
do you have some more?
Yeah, let's get in. Let's see if we can get in one or two more here. Speaking of the teeth and diet and such, our friend Ethan has asked, does wear on the teeth seem to affect the function of the teeth up until senescence? So I suppose as the teeth wear over the age of the animal, does that change how they're functioning?
As the teeth wear, does that change how they function? I know there's a recent study that was published looking at, I can't think of the citation or the first author. I know that Klaus from Europe, he's done a lot of work with like modern ungulates and also proboscideans about their diets. And in that study, they looked at modern like zoo animals of elephants. And they realized that when you had one tooth being replaced by the other one, and you had those two teeth at the same time, the body condition of the animal improved, as opposed to when there was only one single tooth in occlusion. And so it suggesting that in their analysis, they see the cyclical pattern in body condition, that when you have that overlapping, those two teeth, and they relate that to the area, like how much blades you have and the surface area, I guess, the chewing surface area. So that really does suggest that during the age of the animal, an individual animal, as the teeth wear, their efficiency at processing food is not constant. Yeah, it does suggest that there's times when they're more efficient at acquiring nutrients from the food and their body condition is better. They do say, the authors of the study do say that they only see this trend in zoo animals where there's control conditions about what the animals are being fed, because this difference might be quite minute in the wild that, or it may also get lost because animals are eating different plants. So the relationship with the body condition may not be as straightforward as what they have seen with the zoo animals. But I found that very, very interesting too, that to see how, the relationship, how they study the body condition of the elephants and relate that to the tooth replacement, their sequence. Yeah.
We have one more from Greg McDonald, and he is asking about Schreger lines. And one of you might know the answer to this, and that's whether or not you see a difference in this in the tusks in the different species of mammoths.
Oh, I haven't, Chris and I have talked about looking at Schreger lines because a lot of our tusks are fragmentary and they're not complete. And we also want to, we don't know if they're mastodon, some of those tusks. But we haven't done that project, so I don't know.
It might be another avenue.
Chris, you know of like a study, because I haven't read, I know there's differences, of course, between mammoth and mastodon.
Yeah, so I mean, I'll back up a little bit and kind of talk a little bit about Schreger lines. So Schreger lines are these angles. If you look at a cross section of a tusk, they're basically angles between some of the dentin features. And mastodons and mammoths are distinguishable depending on what kind of angle you are looking at in that dentin. I don't know that anybody has looked at it between different species of mammoths. I'm not aware of any. Since they're really reflecting kind of, well, at least we think that they're reflecting larger morphologies of the tusks themselves. If we are seeing very different tusk morphologies, then we would expect to see differences in Schreger lines. But yeah, in Alberta might be a really fun place to look at that because you do have such diversity there. So yeah, please follow up on that and publish that for the rest of us.
You have a lot of fragments, which makes it kind of ideal.
Exactly.
Oh yeah.
David?
And is there any other question? Are there any other questions?
We've got one more question here, and then I think it'll be about time to wrap up. Grant says, my wife is wondering what a hybrid would look like. Colombian with a woolly's poof of hair, or perhaps a balder woolly?
Yeah, what would that look like?
Maybe since of course that's very hard to know, what would you want it to look like?
Yeah, I'm like, what would I want? No, I would want a hybrid with like a little funky pattern of like fur in some areas and then less fur in other areas.
Like a poodle, maybe like a poodle.
Yeah, kind of like
Poodle mammoth.
yes.
All right, we better wrap it up. Anything else, David?
I think that's it. Thanks to everybody for joining us, for asking questions, as always.
Yes, thank you so much for being on the show, Christina. We really enjoyed it.
yeah, thank you so much for the invitation. Thanks. I really enjoyed talking with you and learning more too, because like I said, my main topic is horses. I'm just like sidetracking to mammoths. Yes, for a little bit, and then yeah, I'll go back to horses.
Sidetrack to mammoths. I love it.
Yes.
Most of us get involved, yeah.
All right. Thanks for tuning in, everybody. Thank you again, Christina.
Yes. Thank you. Thanks. Bye.